We are now entering the process of risk governance, and we know that in the first step we do have to conduct pre-assessment of our process to define the entire process, the risk governance process. However, next we proceed to the second step, which is the scientific and technical assessment, and this stage can just be conducted on the basis of reliable data. So we require your data, the data of the scientists, and by tools can evaluate on a scientific basis the risk. We do this under the premises of the FAIR data initiative, and therefore we talk about fairness now and about quality assurance, quality control of data. Nanotechnologies are very important, and they are a huge investment and important for the growth of the European economy. We know that the data on the safety issues for nano are fragmented still, and this is hampering the progress in nano safety assessment. Read-across approaches are currently still not fully available, and therefore Nano Commons project has taken the initiative of trying to collect different data sets, different data formats, trying to harmonize that, and including huge data sets from different projects that they are depicted here in a kind of a way that they can be interoperating with each other. So it's a kind of an e-infrastructure platform to be established to provide a reproducible science, and should enhance the data integration and enabling nanoinformatic workflows to be integrated to in vitro experimentation. This is set upon the initiatives of fairness, data fairness. What does this mean? It's making data findable, accessible, interoperable, and reusable. And this shall, on the long run, remove barriers for regulators as well as for industries. We need to integrate knowledge bases. The Nano Commons knowledge base is one of them, but there are also other ones existing to facilitate grouping and read across for nano safety data. So we need to interconnect our community in a kind of a fair data single market. And by this means, we might then be able to have a much better exploitation of the EU-funded research data. The data landscape, as I was initially saying, is still a fragmented one, to promote data-driven innovation, and that would, on the long run, lead to a very positive socioeconomic impact. So this is somehow, of course, covering the entire data lifecycle. What we are, as experimentalists, used to is doing our experiments and then eventually evaluate our experiments, do some analysis based on the data that we have obtained, in some ways also visualize and use tools for visualization of our data. Establish also the means that other people can, based on this data, conduct predictive toxicity assessment. And then we need to facilitate storage of our data and give online accessibility to that in a fair way. But this is a life cycle. On the basis of this data, other people can work upon. So we need to consider, in order to make this cycle be running all around, how rich our metadata shall be. What is metadata? Data describing our data, and then thus making that understandable and reusable. So we consider the R of the fairness process. So how rich, how shall we collect metadata that we can achieve these high standing goals of providing fair data to the community? Just a few more comments. Data management plans are an urgent necessity to be included in EU projects. To establish data management plans or research outcome management plans, a new term, a new kind of role has been also developed, which is the data shepherd. And you can see here from all the different objectives that might be seen during the data life cycle, from the more kind of acquisition of the data until to the data storage and reapplication for different formats, that many different roles can be defined, and the shepherds is one role that has to have some expertise in many of these, or in actually all of these different topics. So this data shepherd drives this process from the planning and from being integrated already in the planning of the procedure of the experiments, being participating in acquisition, processing, analysis of the data, but all along to storage and fair sharing to drive the cycle, the data life cycle for a profit of each of the other customers. So how can this look like in a very concrete example? One example, you have a research question like, for example, to look at the protein corona formation of different particles with different proteins. And you are interested in, is there some selectivity in binding? How about the orientation of the proteins around, other structural alterations, and so on? This is just our research question. And you would start in your project with the synthesis of the materials, of some surface functionalization. Then you do some proper characterization. You use your knowledge base, like for example, the Nanocommons knowledge base, to upload and provide in a fair means your data. Also make some modelings on your obtained physicochemical characterization, like for example, image analysis to provide further nano descriptors for that. You could then also from this knowledge base, for example, enter to corona modeling to integrate in silico and in vitro data. So in this way, try to establish a kind of an integrated in vitro in silico workflow. And then go along with all your experiments for the functional assays, structural assays that you plan for this project, until you come to the issue of data storage, to do also annotation of ontologies for the different endpoints that you use in a fair way. And this all can be powered by the Nanocommons knowledge base. Okay, so next we're gonna go to the Nanocommons knowledge base, which is provided by the Biomax knowledge portal. We need to click the URL given here on the right upper corner. Okay, we clicked Nanocommons URL and have arrived at the Nanocommons homepage. Here you have access to many things. I would like to highlight shortly the previous events where you have access also to teaching materials and training events that we had performed before. You find also teaching materials also in this user guidance handbook. It's a different way of assembling and finding yourself things along. But today, let's focus on the knowledge base. So here you have the access to the knowledge base. If you click on it, you get access to the portal. What you have to do is registering once, and I have done this, of course, and then you can log in, and I have done this before. This is the landing page. Here you see 4 activities. You can have access to data that are deposited there. Here you can result eventually characterizations or toxicity assays from bigger projects also, and can look up, for example, their metadata templates, which would be an interesting information eventually. You can also perform analyses like, for example, the protein corona analysis, but also TEM image analysis, have access to ontology terms. You can browse ontologies. What we are going to do today, and this is now of interest to us, is data upload. So we can define particles, for example, by creating a new particle in this shell here. You would then be adding the chemical elements of your particles, give some description and a name for your particle. You can also do that by in spreadsheet format by having a whole list of particles to be defined all at once. What you can do here in the upload parameters button, you can also look up parameters that are there. So for instance, if you're interested in ontology terms, you could, for example, look up size. Yeah, and here you get the ontology term for size, for example, or PDI, or the dispersity index, ontology term for such type of parameters. So you look up whether the parameters are already deposited. If yes, then you need not define them. If they are not there yet, you can define your own parameters. But now we go ahead and do upload experiments. So for example, particle characterization, what you need to do here is then to download the Excel template sheet. does this look like? This is basically already such a format. So you need to then input the nanoparticle ID, which you have obtained from the knowledge base. You can define different instances. What is that? For instance, you do define or disperse your particles in medium, in water, or a medium containing proteins, or interacting with soil. This would be different instances of your particle, the history basically in your experimental workflow. And then you define the property, the parameter, like TM or DLS or NTA or something, and give the value with or without the unit. How does this look like? For two model examples here, this is not a real particle idea, so just XXX for one particle and Y for the other one. And you have the properties listed here. We do not have instances, and these are the values for the different properties. Standard deviation of TM and nanoparticle tracking analysis. You can also give input for shape, like the sphere here of a coating. It's an uncoated silica particle versus the other one being an uptest coated, for example. Different values, theta potential values. Okay, and this data set you then upload into the database. How does this then look like? If a real particle, for example, we have a glance on, this particle has been defined here. What you obtain is also the NINCI, so the nano INCI code for that one, a little bit of description, the chemical composition, and for example, some data on this particle in the case of pH 7, for example, buffer. What you also have here, and you can upload the metadata. We can also consult this metadata for creating a template. To include all the parameters that are necessary. So you can here find some examples and have a look into. You would also be able to deposit the raw data, for example, for the and then these values are here also to be seen. And via the knowledge base, you can also, as I said already, perform analysis. As we have now seen in the Nanocommons knowledge base, how to define new nanoparticles, how to look up parameters, how to eventually define new parameters, how to use them, and how to define experiments and upload our data. I would like to finally give just some links to you, for example, the Nanocommons knowledge base, giving you also the access to the knowledge portal. The user guidance handbook I have mentioned, training events, teaching materials are available from there in our infrastructure. You would also find these kind of teaching events, teaching materials at the Elixir tests. Everything has been deposited at Zenodo. There is a Nanocommons and Nano Safety Cluster community there where this material have been deposited. The Nanocommons and Nano Safety Cluster YouTube channels also provides useful information, and I would like to invite you to join the mailing list of the Working Group A on Education, Training and Communication of the Nano Safety Cluster Nano Safety Cluster. If you are in need for further training or interested in further training, teaching events that we are going to offer. With this, I'd like to thank you all for your interest. And I want to acknowledge the funding agencies, which is the Horizon 2020 Research Innovation Program and the Nanocommons and NanoRego projects.
Good afternoon, dear participants. My name is Dmitri Ciornii, and I'm working at Federal Institute for Materials Research and Testing. Currently, I'm working at NanoRego project, and today I will talk about data quality for nanorisk governance. First, I will talk about data quality criteria, which is important for nanorisk governance. Then I will make a brief introduction to technology readiness level. And after this, I will talk in more details about knowledge, information, and data readiness level, KaRL, the approach developed in NanoRego. And this will be followed by conclusions on how KaRL could improve nanorisk governance. To be suitable for nanorisk governance, data need to fulfill certain quality criteria. Some of those quality criteria are represented here. When talking about completeness of studies or data sets, let's say an example, characterization of nanomaterials. Completeness means the availability of complete and relevant physicochemical characterization and also extent to which the experimental setup and methods are described and validated. Relevance is another important criteria, and it is nothing else than fitness for purpose. It indicates the suitability and appropriateness of data for a given purpose. A study can be, for example, relevant for one purpose and irrelevant for another purpose. As an example, in nanotoxicological study, relevance means covering the extent to which data or tests are appropriate for a particular hazard identification or, for example, risk characterization. Another important criteria is fairness. Maybe some of you have heard already about this, representing findability, accessibility, interoperability, and reusability. Fairness puts specific emphasis on enhancing the ability of machines to automatically find and use data, in addition to supporting its reuse by individuals. Fairness allows also machine readability and interoperability between different databases or data repositories and enhances the value of data and studies. Traceability is another very important criteria for data quality, and it means that the data or study or a piece of knowledge is traceable down to its origins, down to the very experiments when talking about scientific data. Traceability is considered as the highest standard for data quality and is necessary, for example, for regulatory purposes. Now I will talk about technology readiness level and knowledge readiness level. Technology Readiness Level, or TRL, has been developed initially by NASA in the 1970s and is a concept for evaluation of the readiness of a particular technology or system. This is a 9-grade scale divided into 3 blocks. First block is TRL 1 to TRL 3, refers to the basic research or lab phase. The second block, TRL 4 to TRL 6, refers to the development phase, and the third block, TRL 7 to TRL 9, refers to deployment. In analogy to TRL, we developed at NaNoRigo Knowledge Readiness Level, KaRL. KaRL is also a 9-grade scale divided into 3 blocks. KaRL 1 to KaRL 4 refers to the technical readiness of knowledge. KaRL 5 to KaRL 6 refers to the knowledge readiness for small scale. And KaRL 7 to KaRL 9 refers to the knowledge readiness for large scale. KaRL 1 represents all available data in digital form or in text files stemming from all possible data sources, which could be databases or physical libraries, electronic lab books, and so on. One example of KaRL 1 could be publications, let's say, on titanium dioxide. KaRL 2 is considered data completed with metadata. And metadata, there are of different types: reference metadata, structural, statistical, bibliographic, or technical metadata. There are also some existing minimum reporting standards, such as Arrive, Miame-Tox, Mirabel, MinChar, Mian. All of those require from scientists to report also different types of metadata. For example, KaRL 2 could be a study which was carried according to MinChar, and MinChar requires the reporting of particle size distribution, agglomeration state, shape of the nanoparticle, functionalization, and so on. KaRL 3 represents data completed with toxicological context, which is very important for regulatory purposes. Recently, in Small, it has been published a nanotox metadata checklist, represented here on the right, which requires from toxicological studies to provide the toxicological data. For example, exposure, acute exposure, chronic exposure, or route of administration, data on toxicokinetics, decision points on hazard identification, and so on and so forth. So the more complete study is with toxicological context, the more suitable it is for regulatory purposes. KaRL 4 represents fitness for purpose of a study. Since there are also different purposes like scientific or regulatory, safe by design, exposure assessment or other, there are different quality scoring systems like Nano Cred, Guide Nano, Klimisch scoring, Daan, and so forth. Klimisch scoring is suitable for studies for regulatory purposes. Here on the right you can see the score grade of Klimisch scoring, and the studies with score 1, which are reliable without restriction, are suitable for regulatory purposes. Another way to score studies is Daan's literature criteria checklist, which requires studies to report physicochemical properties, sample preparation, testing parameters, standard operating procedures, OECD guidelines, and other. In KaRL 5, stakeholders get involved in the process of knowledge readiness. Basically, KaRL 5 defines a list of stakeholder-specific requirements. Let's say a stakeholder is interested in a risk-related question. For example, an industrial producer of, let's say, nanoparticles. It could have a question whether exposure to particles in the working environment is affecting the worker's lungs. So how to obtain the answer to this question? Maybe some occupational exposure to nanoparticles should be investigated, for example, legal requirements for these particular nanoparticles. Also, it could be necessary to provide in vivo data on the effects of nanoparticles on lungs or gather the literature on this data. He could need, for example, combined dosimetry approach or other tools or models for additional information on these nanoparticles. Also, new experiments or assays might be necessary to answer this question. Before we proceed to KaRL 6, I'd like to explain you what is an actionable document. Per definition, an actionable document is a written instrument or document on which an action or defense is founded. This definition comes from military but can be also successfully applied for other areas. And some examples of actionable documents are represented here. It could be a meta-analysis or proposal or scoping review or some dossiers or reports or whatever. Making link to KaRL 5, KaRL 6 represents functional knowledge in a form of an actionable document. You have seen already a few examples of actionable documents. And an example of such document could be a report on toxicity of nanoparticles for an industrial producer. And such actionable documents are normally prepared by third party, for example, a consultancy organization or by a stakeholder himself. Previously, we talked about KaRL 6 representing an actionable document for individual stakeholders, which means that such actionable document is designed for small scale. However, if we intend to produce actionable documents for large scale, for policy, for decision making, regulatory purposes, then we suggest a KaRL 7. KaRL 7 is a level where a specialized governance authority or a group of experts is suggested to perform a quality check of an actionable document. This group of experts could perform, for example, completeness check of all the documents. They could perform traceability assessment, whether the data and studies and knowledge gained until now is traceable. Only then it can be considered suitable for further purposes, for large-scale purposes. So after performing the general quality check of such documents, additionally also broad context need to be included in the actionable document. So KaRL 8 represents a list of requirements for all stakeholders. Here, public, academia, industry, regulator, consultancy, NGOs, and other stakeholders, all stakeholders are involved. Their requirements and needs are combined and analyzed and included in the actionable document. An example of KaRL 8 could be a list of requirements including public opinion, design principles, which is nothing else than social and ethical implementations in nanotechnology, circular economy principles, safe by design principles, good governance principles, or impact assessment. KaRL 9 represents finally the last level of knowledge readiness and represents basically the document including KaRL 1 to 7 and the list of requirements from KaRL 8. Such document represents the basis for regulatory decisions. An example here you can see a opinion of the Committee for Risk Assessment on a dossier proposing harmonized classification and labeling at EU level. This document stems from a CLP process, which I will be talking a bit later about. And if this document is combined with additional public opinions and sign principles and circular economy principles, so all the requirements from KaRL 8, then it can be considered finally KaRL 9 and ready for regulatory decision making, ready for nano-risk governance. Finally, to sum up the KaRL approach, it is divided into 3 blocks, mainly technical readiness, small scale and large scale. And again, to quickly recall each KaRL separately, KaRL 1 is all available data, KaRL 2 is completeness with metadata, KaRL 3 is semantic metadata, KaRL 4 is fitness for purpose, KaRL 5 is stakeholder specific requirements on a small scale, KaRL 6 is a functional knowledge, KaRL 7 is quality check of functional knowledge, KaRL 8 are requirements for risk governance, and KaRL 9 are functional knowledge for risk governance. So how KaRL could improve nano-risk governance? KaRL could support any stakeholder in providing a guidance on how and where to get high-quality data. KaRL helps to obtain reliable knowledge from data and a higher knowledge readiness for different purposes. It provides a list of useful nanoinformatical tools to fill knowledge gaps. It suggests quality check of actionable document by a specialized trusted governance authority. And KaRL also enables involvement of all stakeholders and addresses ethical, social issues, safe by design, circular economy issues, and sustainability. We gratefully acknowledge the European Union's Horizon 2020 for financial support of the project NanoRigo Grant Agreement 814530. Thank you for your attention, and are there any questions? Now, hopefully you all can hear me, and I know that the information about KaRL is fresh, but still I would like to show you some examples of KaRLs. And so next slide, please. Yes, here we will stay a bit longer since here is basically the first attempt to make a KaRL assignment table, since KaRL is still in development and we are still working on it. But based on the information you have heard right now, I summarized on the left-hand side the requirements, and on the right-hand side the KaRLs from 1 to 9. And you can use this table while practicing with me during the exercise. So first, just imaginary case. Imagine we have found a publication on, let's say, zinc oxide, but the section materials and methods is lacking. So my question would be, what KaRL can be assigned to this publication? And since we're lack of time, I would give you, I don't know, 10 to 15 seconds to think about it, even though this exercise particularly is easy. So you can basically write in the discussion forum. Any ideas? Yes, perfect. Thank you, Burkhard. KaRL 1, it is KaRL 1 just because, as all of you know, materials and methods refer to metadata, and since it is lacking, we cannot assign it to KaRL 2. So it has to be KaRL 1 just because data exist, but they are not really complete. Now this is for the warm-up. Next case, we have found a study on Daan platform on toxicological effect of titanium dioxide nanoparticles depending on the surface functionalization and on the diameter of nanoparticles. Now, study was conducted according to OECD guidelines, and here's the question: what KaRL could be assigned to this particular case, according to the table? So, any ideas? Well, I mentioned before that we have found perfect. Yes, thank you very much. Why Dana? Why KaRL 4? Just because we have used Dana platform, we have also toxicological context, and this is a study conducted according to OECD guidelines. Perfect. Thank you very much. I see that it works. Now, second example, a toothpaste producer wants to improve the recipe for the toothpaste. He has concerns about the chronic exposure on titanium dioxide, which is contained, for example, in this product, and the producer hires a third party to obtain needed answers and information to make a decision on the improvement of the product. Third party performs work and gathers all necessary information. And here's the question, what KaRL could be assigned to this situation? It's sad that we cannot communicate out loud, but good that there is a chat. So again, the third party gathered the information on a question of a stakeholder. And yes, so what level could it be?
Maybe you.
Okay, here's seconds more. KaRL 7. Well, KaRL 7 would be too far, but still, thank you for the answers. This is the level where we just gathering information to answer the stakeholders' question. And KaRL 7 is already a quality checked actionable document. And I didn't even mention that we have already a document. We just gathered the information. So this is the whole process. We've found the requirements to answer the question. And this would be KaRL 5, as you can see in the table. And following directly KaRL 5, the question is, third party presented now a short report on toxicological effects of titanium dioxide over a period of 20 years. So now you could easily guess what KaRL this could be. Yes, thanks for the pointer, Martin. Since I don't have control over the screen. So KaRL 5 were just a list of requirements, and then we gathered it and packed it in an actionable document. Okay, maybe some help here. So we would consider this as KaRL 6 because it is the information gathered in KaRL 5 and formulated in an actionable document, which in this case is a short report. Okay, now we can hopefully move further to a more complicated case. A stakeholder is concerned about a certain type of nanoparticles and wants the revision of carcinogenic effect of those nanoparticles on a regulatory level. Basically, he wants European community to classify that nanoparticles as carcinogenic. After several steps of reviewing the information on toxicity and carcinogenity of those nanomaterials from reliable sources, after several rounds of public discussion and other stakeholder discussions, the document containing all necessary information appears on the table of European community, which will take a decision on new classification of this nanomaterial. Also, I mentioned here that such principles as circular economy, safe by design, have also been addressed. So what, KaRL, do you think could be attributed to this situation? Eight. It is very close, since 8 is all of those I said before, but as a list of requirements. And 9 would be the right answer because 9 is already the document containing all of those. So they are basically very close. And as I said, that we are still working on the assignment of KaRL. 8 and 9 is basically the same. So you're right. Thank you very much for the participation. Now, Martin, we can go to the next slide. So now to make KaRL's visualize, KaRL 5, as I mentioned before, is a list of stakeholder specific requirements. Next, thank you. An industrial producer, for example, produces titanium dioxide, and he's interested whether exposure to particles in the working environment is affecting workers' lungs. So this example has been already in the KaRL presentation. And now what do we do to achieve this KaRL 5? Next, please. Now we have a stakeholder with a question. Okay, this is one condition. We can search for all available data. Then we should also check for some legal requirements. It is possible that we might use also some tools or models for this answering this question, or maybe even carry new essays and tests. So now let's go concrete. So we have a stakeholder. What could be next one? What could be the existing data? The existing data could be just in vivo data on the effects of titanium dioxide on lungs. And this existing data we could find from reliable sources. They should be KaRL 1 to 4. Now, occupational exposure to titanium dioxide represents legal requirements. This we need to know the threshold of the concentration of the titanium dioxide, which is allowed per regulation. Next one. New tests, well, it might be necessary also to carry in vitro cytotoxicity tests on titanium dioxide nanoparticles. And also, particularly for this situation, it would be suitable to use also tools. An example of such tool was mentioned before, combined dosimetry or CODO model. So this helps us to estimate the exposure levels corresponding to the concentrations of in vitro. So if we now go to the next slide, exactly, I can show you how it more or less works. So we have gathered some data in vitro. Then we have gathered some existing data from the literature on the in vivo tests. And then we combine it with the CODO model to simulate the exposure concentrations. And from there, we can extrapolate the exposure concentrations for 1 year, for example, for a worker. So this, all these tools and requirements help us to address the question of the stakeholder. Next one. And following this, KaRL 6 would be the formulated document, actionable document, helping the industry, in this case, the industry, to resolve its task. For example, it can be a short report or a table comparing exposure concentrations, or a scheme showing comparison with regulatory requirements. So basically, the answer, this actionable document, helps specific individual stakeholder to solve his need or task. Next one. So, and how can it look? So if we have a short report or a table, so it could look like this. We have gathered some regulatory requirements to know the threshold. We have gathered literature data on in vivo experiments. We have performed in vitro experiments to show, for example, that it's not cytotoxic. And we have extrapolated the exposure levels using CODO model and calculated the exposure for 1 year. So this document would be KaRL 6. But now let's move further to a more complex situation. I wanted to show you just a grasp of CLP for showing you the example of KaRL 9. So CLP is classification, labeling, and packaging, as many of you may already know. And to fulfill the KaRL 9, all levels before need to be achieved. So this is how we define them. Thank you. And here we have, do not be scared of this scheme. It is the whole process of classification of titanium dioxide, which started 2013 by a member state, France, which wanted to put titanium dioxide on the special corrupt list for evaluating or re-evaluating its carcinogenity, so its classification, how should it be classified. And after a series of processes, you can see, which took many years, we had public consultations, we had risk assessment committees, we had meetings of member states and all stakeholders. All of those participations and discussions and comments led to the final decision taken by European Community in 2020 that titanium dioxide should be considered a carcinogenic material type 2. We will come to this slide later. My message here would be that I would assign KaRL 9 to the very last step before the final decision. So having all of those discussions of member states and public involvement, all of those documents gathered from hopefully reliable sources, or must be actually reliable sources, all of those documents which go to the European Community, this is a really high level, this is for regulatory purposes, and that one could be KaRL 9. But as I told you, we are still in the preparation of KaRLs, and when we will publish soon, it will be more defined. So now we can go further to, I don't know, who is the next candidate?
Yes, so since we have talked a lot about data now, and we have seen the KaRL even applied for many or for a couple of different cases, which was very nice. Thanks a lot, Dmitri. We are now looking again into the social aspects, and for this we pull up the recording from Daan, who gives you an introduction to the socio-economic aspects of risk governance.
Hello, my name is Daan Schuurbiers. I'm the director of DPF, a consultancy for responsible innovation in the Netherlands. And I'm a partner in the EU-funded Nano project on risk governance of nanotechnology. And the main point that I would like to make in this presentation is that the effective integration of broader societal considerations is an essential condition for the success of nanotechnology risk governance. Risk governance is more than risk assessment. Decisions to promote, inhibit, or otherwise regulate new nano-enabled products or processes are not fully determined by the quantitatively identifiable risks, but are shaped by a host of wider economic, environmental, and socio-ethical considerations. In other words, scientific evidence is but one piece of the governance puzzle. And to give you an example of what I mean, consider the recent opinion from EFSA on the safety of titanium dioxide as a food additive. Titanium dioxide is used as a food colorant. Its function is to make food more visually appealing. It can be found in things like bakery wares, soups, broths and sauces, salads and sandwich spreads. After reviewing all the relevant available scientific evidence, EFSA concluded that a concern for genotoxicity of titanium dioxide particles cannot be ruled out. And based on this concern, EFSA's experts no longer consider titanium dioxide safe when used as a food additive. So here are a few things to note from the press release. The opinion revises the outcome of EFSA's earlier assessment from 2016. The assessment took into consideration many thousands of studies that have become available since EFSA's previous assessment, including new scientific evidence and data on nanoparticles. And Professor Matthew Wright, the chair of EFSA's working group on E171, said, although the evidence for general toxic effects was not conclusive, on the basis of the new data and strengthened methods, we could not rule out a concern for genotoxicity, and consequently, we could not establish a safe level for daily intake of the food additive. Note that the EFSA opinion stated that a concern for genotoxicity could not be ruled out because of uncertainty around the characterization of E171. This headline in The Guardian, however, concludes that food colouring widely used in the UK is unsafe. And Foodwatch, an NGO which has campaigned against the use of titanium dioxide in food for years, along with several other NGOs, states that the additive E171 is finally recognized as unsafe. The Titanium Dioxide Manufacturers Association, the TDMA, also responded to the EFSA opinion. It stated that the TDMA has fulfilled all data requests from the EFSA and has also contributed extensive new scientific evidence that confirms the safety of E171. So how does this relate to my main point that the effective integration of broader societal consideration is an essential condition for nanotechnology risk governance? The example highlights how the scientific evidence constitutes but one element in the debate on the use of titanium dioxide in food. All the stakeholders try to shape decisions on the use of nanomaterials. And the picture that you see here tries to map the different stakeholders involved in the titanium dioxide case against 4 domains: policy, science, industry, and civil society. In the top left corner, we have stakeholders from the policy domain. This includes the regulators, those European and national authorities that assess and regulate the use of substances. EFSA, of course, plays an important role here, but so do other authorities like the French Agency for Food, Environment and Occupational Health and Safety, ANSES, as well as the Netherlands Food and Consumer Product Safety Authority, the NVWA. And these stakeholders are different from what are called the policymakers, those in the European Commission and in all the national ministries who need to make policy decisions based on the evidence. Note that different departments like economic affairs, health, research, or environmental protection do not necessarily have the same priorities and may advocate different courses of action based on the available evidence. The regulators and policymakers in turn base their information on the evidence provided by science, represented in the top right corner of this image. Science, however, is hardly a monolithic body itself, but instead produces tens of thousands of papers in a wide range of related fields. So which information do we base our opinion on? And how do we determine the quality of the evidence? And what do we do in case of conflicting evidence? Civil society organizations, in the bottom right corner, will also try to shape policy outcomes. Being concerned with food safety and with human and environmental health, they advocate the precautionary principle. If there are uncertainties about the health effects of materials, they shouldn't be used. And industry, in the bottom left corner of this image, is directly affected by the opinions of the regulators and the decisions taken by policymakers. Here again, industry is not a monolithic body, but consists of a wide variety of large companies, SMEs and industry associations representing manufacturers and downstream users. And as the previous slide has showed, these stakeholders will react to the opinions of regulators by offering further scientific evidence or alternative opinions. They may resist a proposed ban on materials that they use in their production processes and may argue that inconclusive evidence is not enough to take such drastic actions as prohibiting certain substances. The point is that risk governance of titanium dioxide doesn't begin and end with EFSA's assessment. The available evidence becomes the playing field of the stakeholders. And as a future risk assessor, you are a player in this game, whether you like it or not. And your objective, neutral data will be used by proponents and opponents to suit their arguments, whether you like it or not. In summary, what the case of titanium dioxide suggests is that, first of all, safety assessment of nanomaterials is an incredibly complex task. And please note that this is just one specific function of a single material. The debate among risk assessors underlines that there are still many uncertainties around the long-term effects of the material on human and environmental health. And these uncertainties do not just apply to titanium dioxide, but to the range of engineered, natural and incidental nanomaterials in many fields of application, from food to consumer products and from paints to textiles. Now, if there are such uncertainties, we cannot simply assume that nanotechnology innovations will all be unqualified blessings. There are many examples of miracle materials from the past that have turned out to cause problems, like asbestos, microplastics, lead, antifoulants, pesticides, and so on and so forth. And we as a society need to make decisions about the use of such materials despite the uncertainty surrounding their effects. And importantly, this is not a purely scientific but a political question. Risk-benefit assessment is a deliberative process that should include the views and concerns from all stakeholders. So this is what I mean with the picture I showed you at the beginning of my presentation. The scientific risk assessment is but one element of the risk governance process that ultimately leads to decisions on the use of nanomaterials. This is not to say that safety is irrelevant. Safety is an essential precondition for public acceptance. We do want regulatory bodies who draw strict lines as to what is and isn't safe, but it is not the only thing that is at stake in governance. In other words, safety is a necessary but not a sufficient condition for effective risk governance. We have to become better at guiding innovation decisions in light of uncertain outcomes. That means weighing the expected risks and benefits of innovation in light of incomplete and uncertain data. As future risk assessors, you can play an invaluable role in helping make those better decisions. It starts with being conscious of the political nature of risk governance, so you can present your evidence not as the final truth that will settle all matters, but as an important piece of the puzzle to guide future decisions.